Yes. We have Dr. Sean Pittock's video up next. So Dr. Sean Pittock is the chair of neurology at Mayo Clinic, and he had led many of the pioneering studies in the similar disease NMO, or neuromyelitis optica spectrum disorder, where we now have highly effective treatments close to a cure of that condition. And he's going to highlight a little bit about the strive for a cure in MOGAD. So can we play the video of Dr. Pittock? He's actually in Ireland right now, as I told you, the Irish mafia. So one of them has left Rochester for a little bit. So
good afternoon, everybody. It's a pleasure to have the opportunity to speak with you during your MOG patient day here at the Mayo Clinic. I'm very sorry that I'm not here to see you, but Owen and John have asked me to say a few words. Just to give you a little background on some of the work that we've done in the past in inflammatory immune-mediated demyelinating disorders. And I wanted to take this opportunity to firstly thank Dr. Flanagan and Dr. Chen for all the work that they've done for MOG, both at Mayo Clinic, but also in the context of the MOG project. Obviously, thanking all of the Center for MS and Autoimmune Neurology coordinators, especially Jessica Sagan and Lisa, who've been instrumental in the organization of this event. And then obviously Julie and Jim from the MOG project. We've been working very closely with them for many, many years. We consider them friends and collaborators. And I really do hope that you've had a good visit to the Mayo Clinic, and hopefully you've had the opportunity to get a flavor of the passion and the people and the work that we do here. We started really on this journey many years ago in the context of neuromyelitis optica spectrum disorder. Remember that many patients with MOG, antibody-associated disease, at one point were diagnosed as having NMOSD, a seronegative form of NMOSD. But this condition, the disorder NMOSD, was really the first in the list of disorders that are antibody mediated and cause demyelinating disease of the brain and the spinal cord. The story of the journey that we had with NMOSD, I hope, will be a similar story for MOGAD. But I did want to just spend a little time just telling you a little bit about the journey that I took with many of my colleagues here over 25 years as we worked on the disease NMOSD and brought it from an antibody discovery to a near cure. Many of the things that we've learned along the way with respect to NMOSD, we're now applying to MOGAD. And Dr. Flanagan and Dr. Chen and his team are working with pharma to run clinical trials with the hope that we can attain a similar result for MOGAD as we have in the past for NMOSD. I was very lucky when I started at the Mayo Clinic in early 2000. I walked into the neuroimmunology laboratory and asked Dr. Lennon if I could work in her lab and do research with her. At that time, a lot of work had been done on NMOSD, which was at that time one of the most severe forms of multiple sclerosis. It was considered part of or a form of multiple sclerosis at that time. And patients with NMOSD had a very poor outcome. 50% of people were blind within 5 years. 50% of people were in a wheelchair within 5 years, and about 30% of people died within 5 years. And it was a very severe form of illness. In Japan, it was considered optic spinal multiple sclerosis. And Claudia Lucanetti had done pathological studies where she found that when she looked at the signature, the immunopathological patterns in biopsies or autopsies of patients that had died from the disease, she noted that there was immunoglobulin deposition, antibody deposition, and also evidence that complement had been activated. And she argued at that time that the disease was probably antibody mediated. Around the same time, in early 2000, Dr. Brian Weinshanker had been studying the benefits of plasmapheresis for that disease and found that patients often really benefited from plasmapheresis. And since plasmapheresis is the removal of antibodies from the blood, the hypothesis that this was an antibody-mediated disease began to grow. Antibodies were then discovered in the neuroimmunology laboratory by Dr. van der Leyen, and that was the result of blood samples being sent by our clinician colleagues to the lab for evaluation. And when Dr. Lennon took the serum of these patients and put them on a composite of mouse brain tissues, she saw a pattern of staining that ultimately was considered NMO-IgG. In other words, the first ever antibody biomarker of an inflammatory brain disease mimicking multiple sclerosis. And that was published in 2004. And I was lucky to have been a co-author on that manuscript. In a year, in 2005, Dr. Lennon and her team, with Dr. Shannon Hinson and Dr. Tom Kreiser and others, discovered what the target of the NMO-IgG was and found that it was a water channel that sits on the outside of the astrocyte. And the astrocyte is a brain cell that makes up about 90% of your brain. Remember, in the brain, you have about 100 billion neural cells, and each of these neural cells are making about 1,000 connections. But astrocytes are a very common cell within the brain. And they're really important in regulating the brain and making sure that the microenvironment is healthy within the brain. It was quite a seismic shift in thinking at the time, because at that time, most research was being done on the oligodendrocyte, which is the cell in the brain that creates the myelin, because remember, we're talking about a demyelinating disease. And so it really was a turn in terms of movement of the field toward the astrocyte and the role of the astrocyte in this disease. So now we had an antibody biomarker, and we found that about 70% of patients with NMOSD had this antibody. And we worked over many years to really try to improve the tests that we did to detect the antibody. And we improved the assay by many fold by moving to a living cell assay, where we actually used live cells that were made to express the water channel on their cell surface. And then we looked for binding of patient antibody to those living cells, and we detected it using a flow cytometry, a computerized method of detection of the staining or the color that was given off by those antibodies as they bound to those living cells. And that now is the best assay we have for the detection of aquaporin-4 antibodies. Now, once we had discovered the antibodies, this gave us an opportunity to try to understand what was the underlying mechanism of the disease. How were the antibodies causing injury in the brain? And one of the things we found during the studies, and again, this was led by Dr. Lennon and her team, was that when the antibodies bound to the water channel on the surface of the astrocyte, a protein in the body called complement was activated. And when complement is activated, it causes a lot of damage. Those activated proteins can embed within the membrane of the living cell and cause holes in the cell and kill the cell. And they also can attract inflammatory cells into the brain. And so in 2010, we thought about the concept of blocking complement activation as a potential treatment for the disease NMOSD. And we, in conjunction with Dr. Wingerchuk, who's the current chair of neurology at the Arizona Mayo Clinic site, we together did a study, and we treated 14 patients with very severe NMOSD with a complement inhibitor called eculizumab. And we had a dramatic effect. We essentially stopped clinical attacks for an entire year for those 14 patients. And because of that spectacular result, we then led a phase 3 trial, which was the PREVENT trial. And that trial showed also a dramatic result. In that trial, we randomized patients to receive their standard of care. Some patients received the drug, eculizumab, on top of their standard of care, and other patients received a placebo. What we found was that the patients that received the drug had a 95% reduction in the likelihood of having a clinical relapse of their NMOSD. And that was considered a dramatic effect. And that actually led to the first FDA-approved drug ever for any form of inflammatory brain disease. And that was the approval of eculizumab for the treatment and prevention of clinical relapses in NMOSD. And subsequently, we've just completed the long-term extension part of a Champions trial, which looks at a different drug, a related drug to eculizumab, but the drug is called ravulizumab. And that drug is administered every 8 weeks rather than every 2 weeks for eculizumab. And we've also had a dramatic result from that trial, where in fact, after 3 years, none of the patients, 0 of the patients that received the drug over 3 years have had a clinical relapse. And that we consider a near cure. And that data is actually going to be presented at ECTRIMS in September in Barcelona and has been selected as one of the best research trials of 2025. So how can we take the knowledge that we've learned from the NMOSD story and apply it to MOGAD so that we can really rapidly move MOGAD forward to a scenario of near cure? And the way we're going to do that is the way that we're currently doing it, with team effort, with close collaboration between all of our researchers, our scientists, our MDs, our coordinators, and of course you, the patients. It's you, the patients, that have the answers within you, and it's the collaboration and our working together that will lead us to making those great strides. The first thing that we've done at Mayo Clinic to really help patients with MOGAD is develop the best test in the world. And that is a living cell-based assay, similar to what we did for the aquaporin 4 antibody test. We use living cells that are expressing the MOG protein to detect the antibodies in patients that have MOGAD, make sure that we have the highest sensitivity and the highest specificity tests. There are many different types of tests out there. Some are very, very poor tests. And we know now from studies that are being done all over the world that the use of the cell-based assays, those kit assays, are not optimal for the detection of MOGAD. And so at the moment, the Mayo Clinic is currently testing thousands of patients every month for MOG antibodies using these really high quality tests. As we study how MOG antibodies are causing or impacting the nervous system in patients, this leads to the identification of potential novel therapeutics. So we know that in MOGAD, interleukin-6 is elevated at the time of clinical relapse. We also recognize that in the context of NMOSD. In fact, another trial in NMOSD of satralizumab also resulted in a significant reduction in the likelihood of clinical relapse. And therefore, because IL-6 was also elevated in MOGAD, a trial is currently ongoing evaluating the potential benefit of satralizumab or an IL-6 receptor inhibitor as a mechanism of stopping clinical relapses in MOGAD. In addition, we also know from our experience with plasmapheresis and other drugs that reduce the antibody levels in patients' bloods that these also can have beneficial effect. And so there is now trials ongoing with rozumab. This is an antibody that has the ability to significantly reduce the amount of antibodies you have in your body. It's an FcN receptor inhibitor, and it has a similar effect to plasmapheresis. So we're hopeful that both of these trials will result in a positive result and allow us to get FDA-approved drugs for patients with MOGAD to prevent future clinical relapses. The field is moving rapidly forward. At the Mayo Clinic, our teams are working on a variety of different immune-mediated disorders, some where the antibodies in the blood are causing the condition, others where T cells in the body are causing destruction of the nervous system. And we're working toward trying to understand the mechanisms by which these things happen and try and identify novel therapeutic approaches that potentially will stop the relapses and hopefully prevent disability as we move forward. These are exciting times, and I'm very happy to inform you and let you know that Dr. Owen Flanagan has now taken over from me as the director of the Center for MS and Autoimmune Neurology. He will be working closely with Dr. Chuck Howe, who's our head of research at the bench, our basic science research, as well as Dr. Oliver Tobin, who will lead the clinical and translational aspects of our MS and CNS demyelinating disease research. These individuals with their teams and their extensive collaborators all throughout the world. We have approximately 30 collaborators in the US, and our collaborations worldwide extend to more than 30 countries and nearly all continents. So we are very passionate and focused on moving the advances forward as rapidly as we can. And we really, really appreciate your joining with us and helping us to do this. So I wish you all the best. I hope you've had a wonderful time and visit at the Mayo Clinic. I'm sorry that I haven't been here to see you all, but we think of you and the illnesses you have, and we work tirelessly towards helping you and your families as we move into the future. So my best wishes to you all, and I look forward to seeing all of the members of the MOG project at future meetings. My kindest regards. Take care.